Welcome back. This is the second batch of recording that I'm doing, and this is hand-to-hand with the journal review that I've just done about OSK, and this is a research and protein. The title is Protein-Protein Interactions, or PPI, and Functional Interplay between OSK and 216 related factors. Excuse me, and this 216 related factors are actually just downstream targets of OSK. Okay, and this is the key figure, and on the right side is a string. The string is the well-known database which collects information about the physical interactions or functional interactions of the proteins. And here is a cluster or complex which contains OSK, our target of interest. And the POU5F1 is the just alt name of OCT4. And here you can see OSK, and there are two other factors, NANOC and SAL4, and they form a physical interaction with each other. So the nodes are just the protein names, and the edge is a degree of confidence. And the interesting thing is that they actually form an interchangeable complex with each other, but also MIC, another factor of Yamanaka, is not included in this set. So the five proteins physically interact with each other with high degree of confidence, but MIC is not. And there's another factor called, I'm obstructing over here, the Lin28A behind this my face, are excluded from this niche of interactions. Okay, and this is actually the full picture of the, so you can actually trigger the option in the string, so you can show the physical as well as functional. So functional is the more broader term to show you the network, and this is a functional cluster, and you can actually see when I put 216 of them, cluster 1 and cluster 2. Okay, so cluster 1 were centered by the this hexagonal network by OSK, NANOC, SAL4, and Lin28, but not MEC. MEC was excluded from here, from this functional cluster. And another cluster that I could have found was centered by BMP4, which stands for bone morphogenic factor 4. So functionally, there are two important clusters from 216 plus OSK factors. Okay, and again, I've reduced it into the physical interactions only. So before I've showed you functional, and now I'm showing you physical. And then in terms of physical, it's the same slide, same figure as the key slide. But over here for BMP4, I've lost many interactions. So cluster 1 has high evidence of forming an interchangeable physical complex when functioning, while cluster 2 does not. Okay, so that's another finding. And the finding about OSK playing as an interchangeable complex is actually supported by previous publications. Over here, the table provided by Zhang et al. In 2020, you can see actually OCT4 SOX2 forms a dimer or trimer, and OCT4 SAL4 forms a complex as well. And OSK together forms a trimer, or I don't know, more, a quaternary complex, for example, to function a native expression. So in terms of the cancer stem cells, it is already known that these OSK factors are working together to have a common function, especially about maintaining stemness and the development of the cells. And this is supported by the enrichment study. So the y-axis is the GO term, and the x-axis is just plain gene ratio. And the color, more darker, the green is, it's about the higher confidence. And you can see the functions over here are actually stemness, development, and transcription. When I put in the input as the five proteins, the OSK plus SAL4 and NANOC in terms of enrichment. Yeah, and that was it. And that was it for today's research. And this is the later slides are about the appendix. So I actually wanted to look at what the function, the cluster of 1 and the function of cluster 1 and cluster 2 is as well. And it's broadly similar. So for the functional portions of cluster 1, we could see a lot of things like That's already shown. But interestingly, okay, stemness, development, transcription, and metabolism. But metabolism was the interesting thing that I wanted to make an emphasis of. You can see that for Lin28 and Lin20B, they actually function in microRNA catabolic process, and I think this is an important finding. And you can see over here again the OCT4 and the Lin28 and this TRIM 71, which is another important factors to maintain stemness, is actually involved in regulation of gene silencing by miRNA. So it is important that you actually the metabolism and the transcription goes hand to hand to actually regulate your young stage. So maybe miRNAs and small RNAs are important for aging. I don't know, probably. And cluster two. This is interesting as well. So it only has small number of genes. Well, it contains, for example, BMP4 and SHH, which stands for Sonic the Hedgehog gene. Interesting, isn't it? Anyways, it's involved in development, solely development, proliferation and development. That's interesting as well. So it's slightly a different function from OSK, slightly, ever so slightly, but I think in my opinion, OSK is more upstream, and BMP4 and other these kind of morphogenic factors are downstream of them. Maybe these are important or not, I don't know. But anyways, that was the appendix. With my full just guessing work. Okay. Well, shouldn't gibberish. Anyways, bye bye.